but thank you for joining our webinar. It's our pleasure to host Professor Ray D'Agostine from the University of Melbourne, Faculty of Engineering and IT, but also as the co-founder and I think Chief Scientific Officer of Tiny Bright Things, who's going to take us through an introduction to the Halo microscopy application. Ray's going to give a short presentation. I think there'll be time at the end for questions. And I think the plan is that they're going to set the microscope up in the BOMP medical facility node. You guys will get a link after this and also in the chat during this meeting to book in a time if you want to come and bring some samples and test out the system. But yeah, over to you, Ray, and looking forward to hearing how it goes.
Thank you. Thank you so much for letting me speak today. Before I get going, I wanted to acknowledge the traditional owners of the unceded lands on which we work. I know it's a webinar, but I think we're all pretty much in the Parkville canvas. So this would be the Wurundjeri, Wurrung, and Burrung peoples. We pay respect to their elders past, present, and future. Let me share my screen. And then we'll get going. And thank you, Paul. As Paul mentioned, I'm a professor in chemical engineering, but also part of Tiny Bright Things. And while this was developed by out of work from Melbourne Uni, it's now translated into a product commercialized through Tiny Bright Things. And I'm here today to talk to you about what it can do. But because of the audience, and this is still a technology finding its feet, part of today will be trying to show you examples to motivate your own interest. And as Paul mentioned, that's why we're setting up a HaloElement in the MDHS site of BOMP. So one of the things when you look at imaging, you kind of go, do you really need another microscopy method? I mean, you've got confocal. Light sheet's been a lot of fun. It's fast. It can do bigger things than confocal sometimes. There's lots of structured illumination. For people that do life science fluorescence or confocal imaging or histological staining, you're really good at it. You've got set up workflows. Why change? And that can be true of other methods as well, SEM or TEM, if you're trying to get an image of something as well. But even now, there's still challenges that will come up. You might have things that are delicate, or maybe they're beam sensitive. In the label-free space, sometimes you can't tag or fluorescently dye what you want to do, what you want to see, because it could kill it, hurt it, harm it, or if it's a dynamic process, it's kind of hard to see it when you label it. And you can get complex morphologies. And so, when we think about microscopy, people often talk about imaging, but really, I think what we're really trying to get at is insight. And we're just using these tools to better understand what we're looking at as our sample. And so that brings us to the HaloElement, which is an attachment that sits on an upright microscope. I've got the same similar kind of capability slide area, but now I've got kind of where the HaloMicroscope can operate, and those are all HaloImages there. But while we invented some of this technology on a half-ton optical table, we've miniaturized it to something that'll sit on an upright microscope. It uses LEDs, not lasers. It uses a sequence of them to build an image. You can get submicron and nano detection, no moving parts. It'll work with aqueous samples or dry ones. It effectively does edge tracing, and it works as fast as you can take a picture, and it can even go to higher frame rates. So that's an interesting way to look at samples that maybe you couldn't before, or you're looking at them a little differently. So what I'm going to do is explain a little bit about how it works. I'm going to say exactly what it does, and then I'm going to show you it actually does different things than that. And we were surprised ourselves, but we're really trying to reach out to people to hear how it might work with their samples. So what we use is called, we use a total internal reflection of visible light. So those of you that work with turf probably know what total internal reflection is, but there you're feeding in one wavelength and exciting another. This is not fluorescence. This is just using visible light. And what we do is we have a circular slide, which we use as a waveguide. and we feed in an evanescent wave. We actually feed in 15 different evanescent waves, kind of like on a clock face around a circular slide and the samples actually scatter that evanescent wave. And from that, we can see a sample. So it looks kind of like a dark field image. Now, for simple samples, it actually does edge detection. And you'll see in the, let me get my laser pointer or my pen, actually. It's always, you'll see over here, we've got simple samples. We actually get the outline in these little dots, and those dots are effectively scattering hotspots. So this is just a picture of a bright field image with a particle being scattering one evanescent wave. And what happens is those hotspots, which are a result of the particle frustrating that evanescent wave and getting some scattering that goes up, those hotspots are sometimes called resonant hotspots, but they actually end up on the edge of the particle. So on the right, next to that schematic, we have a bunch of 5-micron polystyrene particles, and you can see there's little dots there. And I'll get to why it's a rainbow color scheme in a moment, but those little dots are hot spots from those particles scattering 15 different evanescent waves, and we put it all in one image together so you can get the outline of the object. And so it kind of looks like a dark field technique. But we're using evanescent waves to scatter light. And so the particle literally gets in the way of the evanescent wave. But what it means, though, is evanescent waves are not diffraction limited the same way regular plane wave light is through a microscope. So you can scatter on objects much smaller than the diffraction limit. And in some cases, you can even get measures of their size, but you can detect things that are incredibly small compared to a regular microscope. Now, the fact these hotspots appear on the edges of things actually is kind of something that we've learned for simple objects. HaloMicroscopy is really good at edge detection. And so it's a little bit different than how you think of images normally. But before we get to that part, this is what it looks like. This is how it works. We have just 15 red LEDs going around in a circle. We're triggering them sequentially. But if you look through the microscope, and this is a field of view with just some 5 or 10 micron polystyrene particles, what you'll see is sequentially we record those hotspots that go around the particle, then we can composite them together. Here we even went through and found the specific quantitative locations of their centers, and then you can even group them together as well. And so it's really good at seeing the edges, but it's red light. This is a red wavelength. There's no fluorescence there. Now, because there's a little bit more information in the fact that we have lights that are located on a clock face, their location for a sphere doesn't really matter that much. But if it's an anisotropic object or complex structure, the location of the sample with respect to the light actually can have more information in it. I'll show you an example a little later where that's the case. But it does mean when we take our 15 images, we give each image in the sequence a different color. And then the final halo image is we composite all of those 15 images, we stack them together, and that's actually where that kind of rainbow color scheme comes from. Technically, it's not rainbow, it's a basic color scheme called phase. And you can even download it in a MATLAB library if you want. So these composite images, we stack them together and we get a composite image of those 15 images, and that's what we call a halo image. Now, as I mentioned, it's good at detecting the edge. And so this sample is actually a 50 nanometer film. We did do the Melbourne University logo as well. This just happens to be the Tiny Bright Things one. And if you stare a lot at the halo image above, below is just the bright field image. If you stare at that, you'll notice inside the letters looks really dark even compared to the background. And that's because we're illuminating the edges really well, but not so much on the inside, because that's an isotropic, same material, no features, no cracks, where we're just really picking up the edges, not necessarily the inside. Now, this is a gold sample, so we made it on glass and we used EBL to pattern it. Now, we made a couple of these samples, but we accidentally left one out in lab for 2 years. So if you don't take care of your EBL sample and let it sit there for EBL made gold film and let it sit there for 2 years, it can get cracks and start to maybe delaminate. And so this is one of the other logo samples where we were terrible to it for 2 years. And there you can actually start to see that we got cracks in the film, and it was really good at illuminating those cracks and defects within the film. It's finding edges, but now it's finding edges inside of a film, and also the letters as well. But you're really sensitive to edge detection, but it doesn't have to be in a gold film. Uh, I think some of you will be more familiar with this sample than I am. It's a Lilly section, kind of a standard microscopy sample where you can see their bright field is below, but the halo image is really good about picking up the edges. Now, I'm going to talk about mostly biological samples, but I'm going to show you a couple materials ones just to try to explain a couple things. So a metal organic framework is just that. It's a material that's a mixture of metals and organic ligands, and these things assemble into crystals and amorphous materials. They're often used as membranes, or they're often made into particles. They have a lot of internal structure, but they have crystalline regions and they have amorphous regions. And this is just a big moth particle that happens to be triangular. But you'll notice there's scattering on the outside and kind of this region where it's scattering strongly, and on the inside you see a darker triangle. And the difference is this material can exist both in a crystalline state and an amorphous state. So one's very ordered, one's kind of messy like a glass. And so what we're doing is we're scattering off the amorphous state on the outside very strongly, and we're detecting defects in the crystalline state, which is dark, where those colors are defects in the crystalline state where it's become amorphous. And that's because we're very sensitive to scattering off two things: geometry and boundaries, but also simply changes in refractive index. And what we find is you don't need such a big change in refractive index, or the change in refractive index can actually give you really resolvable scattering, even if it's a small, thin film, or it doesn't have to be as thick as you'd expect. And I'll show you a fun example in biology. But the other thing I want to point out is we're going to just show you a particle. Now, this is silicon carbide. The folks in minerals processing have particles laying around, and I'm in the chemical engineering department. So that's a terrible bright field image of it. And then of the same image, there's a halo image. But this is a little bit different. These are particles that don't just have rough edges, they have internal structure and grain boundaries. And what we're seeing in this little inset zoom is we're actually starting to see the internal structure of the object as well. So you'd never see that in an SEM because that's a surface sensitive technique, and it's kind of hard to see in bright field. And I'm not quite sure how you would do fluorescence on this. Maybe you could try XRF, but I don't know if you get the resolution for XRF imaging. But what you're seeing is internal structure. And that gets kind of interesting because when we realized we started to see internal structure and we could do a little bit more than finding the edge of simple objects, it made us think about why. And we spent a lot of time looking at detailed optics modeling, which I'm not going to go into today, but I did want to illustrate it with a very simple ray optics model. So if this is just assuming all the light is a bunch of rays and just tracing through how they diffract and refract and reflect at interfaces. So we're taking total internal reflection. And this picture is a single particle where we've turned the bright field off, and you just see the resonant hotspot. And then these are reminders. But over on the left, we have an evanescent wave down at the bottom, and this is, say, a 10-micron particle. And so it tells you two things right away. One, ray optics isn't always that. It's a little bit more complicated to look at all those rays bouncing around inside a particle. The particle doesn't have to be in that much of the evanescent wave. It only frustrates it or scatters it here, but it's because the light bounces around inside it. One could even say resonates inside the particle, that you actually start to illuminate the internal part of the particle. It's this resonance that also gives you the hot spots on the edges. And this is just another view where the evanescent wave is down there. So the evanescent wave maybe goes 500 nanometers into the surface, maybe a micron, but you're actually able to illuminate structures much larger than that. But why does it scatter things on the inside, like the internal structure in the silicon carbide? So imagine for a second that that particle had an occlusion. Could be a void, maybe a different region, a crack even. Well, what's going to happen there is the light bouncing around and resonating inside the object is also going to scatter off those occlusions in internal structure. And so that allows you to see perhaps things you didn't expect. Instead of finding just edges on the outside of an object, you can find the edges on the inside of an object. Now, with that in mind, what I'd like to do is take you through some examples of how objects can frustrate or scatter an evanescent wave, and you can illuminate a very large set of objects, and then show you how we can look at internal structure, but less on particles and more on biological samples. So the first one I'm going to talk about is how we're trying to look at Halo sometimes, or how the HaloElement works for customers. Is how does it fit in a workflow you already have? So the first one is fibers for cell culture. And this is an example of SEM fibers that are electrospun fibers, and this group typically uses SEM first before they actually start to try to use them for tissue culture or cell culture to characterize their fibers before they're spun. And so the workflow then is EM and then further microscopy with cells when they're wet. Now you might have to worry about beam sensitivity here, or you might have to code it. So not that sample, but a similar sample made by this group. This is a HaloElement image of similarly spun electron fibers. But this could be in air. This also can be in water. This can also be with the cells. And you see a couple things. First, this is a really thick focal block. And you'll see some of it almost looks a little out of focus. And you're actually illuminating the whole focal block because that evanescent light wave actually couples into those fibers, and the fibers effectively work as a light pipe. So you end up illuminating the entire sample, even though the evanescent wave's kind of a small fraction at the bottom. This is a much thicker sample. Now, in this instance, maybe you don't want to do an SEM if you want a quick check on your fibers. Maybe you want to make sure that they're worth taking the time to put in the SEM, or maybe you want to know right away, you just made them, you throw them on a slide and look at it. Or maybe you want to start looking in a liquid or throw a cell in. This changes how you might think about that workflow. That's a suggestion or hypothesis, looking for people to test. Now, another example for a more natural fiber is these are collagen fibers, and I'll admit that's not a great bright field image, but this is the Halo image where those individual filaments are kind of 200 to 300 nanometers, and this is just collagen in water spread on a slide. And normally this group ends up drying it out, doing an SEM to make sure the collagen fibers are there, and then has a parallel slide where they actually start using it for tissue culture. So again, for that fast check for seeing samples that are really hard, unless you need to go into EM or come up with a staining protocol, this has potential to be interesting. Now, let's talk about cells, because so far we pointed out we're doing imaging, but we're just using visible light. There's no fluorescence, there's no labeling or staining. And so these are muscle cells. These are fixed muscle cells that was actually set up as a confocal sample, but this is just illuminating them with red light. There's no, this is a red wavelength. There's no fluorescence there. You can see the nuclei. You can see where the membrane is. You can see cytoskeleton as well. You can even see things in the nuclei. And it's different in that you're actually seeing the whole cell as it is in nature, not just the parts that you might tag in fluorescence. But it's a really different approach because now you can start to do label-free imaging, and you can even see where the membrane is. And remember, the membrane's not that thick, but it's a really big refractive index difference compared to water or the background fluid. And so that's why you get the contrast and you can see the cell walls. Now, the next example, I know no one would probably say, wow, I desperately don't need to image yeast. And that's true. Everybody knows what yeast looks like. The reason we took a picture of it was because everybody knows what yeast looks like. And this is just in media, live yeast. And on that zoom in, why I point that out is we actually watched in real time, and we just kept taking screenshots as we watched those yeast rotate around and the things inside move around as well. And that's just a way to calibrate your sense of what you can see for something that's known. Now, instead of muscle cells, you have to ask yourself, where would this be useful? And this does get back to workflow. Bright field's not fantastic, but would you use a halo image before you stain? Remember, it takes longer normally to focus on the sample than it does to take the image in halo, whereas this is a gorgeous piece of staining work done by the same group that supplied this sample. And as you can see, gorgeous staining confocal image, but that takes a bit longer to do. So I'm not saying I would wonder if researchers would ever, I don't think they're ever going to replace fluorescence and confocal, but you could see where this might help in fast checks on samples or maybe whittling it down to the ones that you actually need to go actually do on a confocal instrument. And it's a lot less work and training to get the Halo image. We can also look at cells. We've looked at both fixed and live cancer cells from different groups. This gets a little interesting. It's certainly pushing my limits of biology, but I'm told these little connections to the surface are difficult to see. This was just interesting. Not only is the cell stretched out, and you can see the pill at the ends, but that kind of background network, I think, is the polylysine that was on the surface that kind of aggregated that the cell stuck to. This is just a different cancer cell where you can start to see they're stretched out and you're moving for mobility. We have some application notes that delve a little bit more into some of the features, but I honestly didn't want to tell this audience about features for cells because I felt like you'd be telling me. But it's interesting when you start to look at a host cell, the types of things you can identify even without fluorescence. So in a sense, cells are large. You can see the evanescent wave penetrates, illuminates the whole cell, even though it could be several microns tall. But we also talked to immunologists, and the first one was this was great. We talked to them. They said, "Hey, could we look at a toxic fungi?" And at the time, I went, "Well, why don't we bring it to your lab instead of you bringing the toxic fungi to my lab?" But this is just from what I understand, a reasonably common fungus, and you can start to see both the cell wall or the wall and a lot of the internals, and again, or the vacuoles. This is label free, and we're starting to get to objects that are a little smaller. Then we, of course, started looking at bacteria. So this is E. coli soup, and what I say that is this was obviously too high of a concentration, but this was broth to slide in less than a minute. We put it on a slide, put a cover slip on it, and just took a picture. The colors here are a little bit from their orientation with respect to each other and their light sources, which is why they look like they have different colors. And you can start to see E. coli, you could get different shaped bacteria. This immunology group was very interested about looking at what happens when you start to make things die and they spit out outer membrane vesicles. One other thing, though, if you zoom in on some of these, because E. coli are kind of like a particle, but they're not, they're double walled. But you can actually start to use edge detection on these as well, where you can start to use those hot spots to actually even try to get a little bit more information about their shape. But it comes back down to, again, workflow, dependence, and the detail you want. At least that's the way I'm seeing it. That E. coli, if you want to get an image in less than a minute, see where they are, look at other bacteria, maybe see their life stage, or maybe you're going to expose them to something that's going to start to degrade their membranes. This gets pretty interesting. Now, the same group that provided the E. coli that we were doing the test with also have published gorgeous confocal images. That's also wild type E. coli. And they've even done some cryo TEM work to get really high resolution detail of the bacterial wall. So it doesn't, we already know not every instrument solves every problem, but where HaloElement might supercharge or fit into a workflow gets pretty interesting about saving time or exploratory work, or maybe deciding what image do I need? Do I need the gorgeous confocal one and I got to carve out a couple days, or can I get it in 5 minutes? And I don't know the right answer to that, of course. I'm not an immunologist. The last few things are a little bit more fun. What we found is it doesn't always have to be about small stuff, it's about detail. And so I have flora and fauna. And the first one is a lily stems tissue section again. That's not a bad bright field image. And that's the halo image of the same section. And you can see you see the edges more clearly. You start to get to more structure, and then we thought, well, what can we do a little bit better? So we zoomed into one region and then took that sections at different focal planes. So we actually did our stepping. We didn't have a motorized stage, so we did it manually. And so that's the type of detail you can get into. This is a plant section. When you stack a couple focal blocks together, and this is just the Z projection of that. And so that gets interesting about what type of detail you might look for in these are larger samples. The next example has really got me wondering. So this is a live C. elegans worm, and this is a juvenile, which is why we're still able to see it at I think this was a 20x objective. I mean, in the next frame, it actually moved its head. which kind of, well, it surprised the engineers. The biologists went, well, that's a good sign. But what you're really getting is a lot of different detail at a lower magnification. And we started to look at these structures, and then we wanted to compare it to something. And then we learned that there's a website called Worm Atlas, and I was happy to know that was in the world. But it starts to help you see that you might be able to illuminate things at a lower scale that are more interesting, perhaps capturing more detail. There's no reason this actually can't mount on a stereoscope. We're really curious about some things about looking at this, can it help in IVF or ICSI or things like that? Or looking at lower magnifications than say a 40x, where you're actually getting a lot more detail even at a lower magnification. And so this is not exactly live flora and fauna. This is another section, but now this is human kidney. And this is a liver section, sorry, not kidney, liver. This is the liver section where the liver has hemochromatosis. And this is, we've been working with Chris Freelance, who runs the histology platform. And we're actually exploring a couple things about how Halo might help identify, change a histology workflow. So on the top, we've got a HaloMicroscopy image of an untreated paraffin section, a dewaxed one, and then this is a pearl stain for the iron. They also did H&E, but I left that one out. So in the bottom is the bright field images. That's what you would normally look at in histology, right? You've got the untreated paraffin, then you've got it dewaxed, but you really need that pearl stain to actually figure out where the iron is. But two images here are going to kind of wiggle. And that's the first one in the halo and the last one in the staining procedure. And so what you're seeing is the regions that show up in the final staining procedure start to show up in the untreated paraffin one or the dewax section. So I would never suggest that people that do histology staining get rid of their staining. There is a lot of power that comes into that in pathology and in research. And I've had this conversation with Ali. He's never given up staining. He wants more lasers and more wavelengths to look at his tissue sections. But when you get to samples where you don't have a lot of it, you want to get more insight before you have a staining pathway, this becomes an interesting idea. And one of the other things we're working on exploring is stacking tissue sections as well, as something Chris and we're interested in working with Chris Freelance to see how this might fit as a tool to complement or fit into some workflows where maybe it's a little bit harder to do staining. Or, you know, the crazy one was, could we do this where you could slice in theater and look at something quickly? And so we're still nutting out that potential. So we're looking for ideas in that. And all right, so the last few things, because I want to leave time for questions. You know, the regular acquisition takes less than a second. It works at about 60 frames a second. And this was one where these are live in water. This is bull sperm. This was a project between Faculty of Science and Faculty of Engineering. They were looking at the fluid mechanics of the flagella, which apparently are very difficult to see in bright field. And clearly they show up in HaloImages. And what was interesting is the flagella and the sperm only needed to be near the surface, and it would light pipe down the flagella, even if the whole flagella wasn't in the evanescent wave. But what we also did is if you go down and take a smaller image, you can acquire at faster rates. So I made a little movie. This was a smaller part of the image, and we're looking at sperm. This is 200 frames a second. It's a rough movie, and part of it was this was the initial work, and it's just way too many objects in the field of view. Why I think they were a little interested was they actually do this in a flow stagnation point and immobilize the sperm either with fluid or chemically and then watch the flagella move. So this is an example where you can look at something dynamic and in real time. And so if you're curious about materials, please ask me. I have lots of other material stories. But the last two things I wanted to point out, because I think these are important for biology, is I've kind of said, hey, we use a round glass slide. And I'm sure people in the room going, I would never use a round glass slide to look at my samples. I might use a Petri dish, I might use a well plate, but why would I use a round glass slide? So I did want to just talk a second about sample handling. That's the HaloElement with a round glass slide. You can put your sample straight on there. You can put your sample on there, a drop of water, and a cover slip if you wanted as well. But we actually found it's probably easier. We've gotten this feedback, and is we made a little plate to hold the round glass slide in place. You put that on there, and now this is a standard microscope holder. So then you can have put a microscope slide on there, and you can put one after the other. For that to work, you put a drop of matching fluid, although sometimes I do admit we use water to optically couple the two together. And what I mean by that is after you put that slide on, then you've got a slide on, the halo will work just fine, and you can swap slides out one after another. And the optical coupling part is just because we're using a waveguide, if you optically couple something to the round glass slide, namely a microscope slide or a petri dish, or to be honest, I'm not going to show you the data, even a silicon wafer, you can couple the light into that object your sample's on, and then you can use microscope slides. And you can put a cover slip on there. You can do things wet or dry. But not everyone uses a microscope slip. You're going, wait, slide or a cover slip. I have a cell. It's not fixed. What do I do? We'll take a petri dish. We really like ones that are flat and have optical glass on the bottom. We actually have a couple preferences. We even built a little holder, and you can throw again, matching fluid. Water isn't a perfect match, but it works pretty well too. And then you've got an optically coupled petri dish on there, and you can look from above. Now, we say you should use a cover slip, but last week I was looking at live stem cells, and we were seeing things just fine looking through water at 10 and 20x. The cover slip, at least for me, was helpful at 40x, so I didn't get my non-water dipping objective in there. You could, I suppose, use a water dipping objective, but we just actually got pretty good results with the 20x. And of course, what was different was you need to keep things warm, so we've integrated an off-the-shelf IVF heater, and we have a holder that'll hold that in place, and you can throw your petri dish on top of that. So we can keep things warm as well, because the stem cells I was looking at last week were 37C. At 37C. And, we only looked at them for an hour or two, so we kept them warm. Obviously, we didn't have an incubator in there, gases in there as well, but at least we thought warm was the first good step. So that's just a little bit about sample handling, because sometimes we get those questions. But really, after all this, where we are is we need your help. We want to find out where this could be more useful and what types of biological samples you could use that this might save you time. And so we have a HaloElement coming to BOMP, as Paul mentioned. It's going to be limited time only. We're going to leave there for about a month. And Dr. Tongwa Das, who's our applications and product developer, who is the world expert on running samples on a Halo, is going to be there for about 4 weeks. And she has a calendar invite. There's a link for it in the chat, and come and try out your sample. We'll have a collection of our the petri dishes we like if you want to try those out. If you do come with your own petri dish, flat glass one, no ridges on it. But yeah, we really actually want you guys to come try things. If you get a picture that's useful, great. And we want to hear your impressions. So with that, thanks so much, and we'd be happy to take any questions.